So I'm going to continue now talking about mining the mixture, and I'm going to talk more about what happens in courtrooms and how we explain these ideas to juries. So what you would see first in a courtroom is that there's a TrueAllele report. Oh, you might see a request for funding for a TrueAllele report, but typically your starting point is a TrueAllele report, and it has various background information, describes the evidence, the methods used, and then by the second page there are match statistics. It will say the computer considered 3 or 4 items of evidence, we ran it, this is what we did, and then there'll be a match statistic. And in blue is the match statistic that we report. And it reads like this: a match between the evidence and the defendant is a quintillion times more probable than coincidence. I think it took about 5 years to come up with that language. There's 12 ways I know of how to write this likelihood ratio match statistic. They all give the same number. Some are very understandable. I think this has plain English in it. Jurors know what the words mean. It's all correct. And then there are others that are not that understandable. They involve a concept called a likelihood that's difficult for many scientists. It's not actually a probability as we think of it. It's a probability of an event given changing parameters. And most jurors don't know conditional probability or likelihoods, and then the statistic they hear phrased is a ratio of likelihoods. Very British. That's where it came from. And I don't know that jurors or normal people, or even most scientists, follow this. But the same statement would read in this less immediately appreciated way as: the probability of the evidence given the prosecution's hypothesis is a quintillion times more than given the defense hypothesis. That's pretty abstract. So I'm not going to go into that today. I can talk about it. I've lectured on it. I have videos on our website and things I've written. But suffice it to say, there are ways of making the statement that a juror might understand and ways that might be confusing. We don't just give a report, we also have a disclosure packet, depending on the number of items. It might be anywhere from 100 to 500 pages. We document in a actual physical document the case notes, the data that we use, the pictures of the data, the questions we asked to the computer, the genotypes that it inferred, what we compared with, the populations, and then the match results and error rates. We're very big on reporting error rates. This evolved over time. My first case, I think I walked in with 1 sheet of paper and people kept asking more and more questions, so we gave more and more. Finally, we just said, look, here's a DVD. It's got everything on it that we can think of. If you think of anything else, let us know. And that really closed off a lot of strange questions on cross-exam of why didn't you provide this, why didn't you provide that, to the point where someone was once trying to persuade a judge that everything that we had on the computer, if it were printed out, would be a wall full of volumes, but didn't mention the computer and was intimating that we were hiding a wall full of paper. So by providing the software for review along with the files needed, if the other side wants to deforest their own neighborhood, they can by printing everything up. But we like giving all the information. I'll come back to these 12 binders, which is part of the disclosure later on. So I thought it'd be helpful to talk about what we show a jury. We also show this as the first part of an admissibility hearing to educate judges about the method and how it works. This was in front of Judge Michael Kukoma in Schenectady, New York. And I'm not going to present it to you the way I would at trial, because that would take 45 minutes. I don't want to spend more than 15 minutes on it. So I'll describe what's here. If you read some of the material about DNA identification for scientists, or just look on our website, or invite me back if that's what you want to hear, like do a mock trial, whatever you'd like. I can go into more detail. But I want to tell you about what it is that we cover. In fact, there's a video of a CLE where this is all I'm talking about for 45 minutes that we do as part of standard trial prep for a litigator. So I could forward that link to you as well. We begin by talking about DNA biology. What is a cell? The human body has trillions of these cells, and most of them have a nucleus with a full copy of the human genome from a person's mother and their father, all in that nucleus. It's chopped up into 23 chromosomes, so a person has 23 pairs of chromosomes, and if you look at a chromosome and you unravel it, what you see is text. So what we're really looking at is text, represented in a biological alphabet using a biological encoding system of the DNA. And we're looking at particular markers called short tandem repeats. The most important concept of them is they were chosen because they have a lot of variation between different people. And so they can be used for distinguishing people from each other. If you choose a marker that doesn't distinguish people, it's not that good for forensics or genetics. This is a metaphor we often use of an encyclopedia having 23 volumes corresponding to chromosomes. And scattered throughout this encyclopedia, there are about 100,000 paragraphs. Each of them is a short tandem repeat of different kinds. And the basic way a short tandem repeat is constructed is there's a bunch of fixed DNA text. This is written out in English, just to show the concept. Starts off, take me out to the ball game. Then it continues. And then in the center, you see, let me root. That word root, shown in blue, is repeated a different number of times in different people on different copies of the DNA. So at this location of what you inherited, say on a particular spot on chromosome 11 from one of your parents, it's all the same paragraph, but that paragraph is different from most other people. There may be 15 different lengths. The word root could be repeated 7 times or 11 times or 18 times. And because it's repeated a different number of times, the length of the paragraph is different. The more repeats, the longer the paragraph. That length variation is what scientists are measuring as their genetic variants that are called alleles. The most important concept in genetic identification is the genotype. The genotype is the pair of alleles that a person has at some location on a chromosome. It's also used to refer to, if you're looking at 20 locations, what are all 20 pairs of alleles. But it's a pair. You have one from each parent, as shown on the bottom left. You have, typically they're different lengths. It could be the same, a length of 10 repeats, another length of 10 repeats. In this case, I'm going to use the example of a 10 and a 12. The genotypes are really what we're looking for. The alleles are more like the bricks that make the building. The genotypes are the building. And those genotypes have a lot more variation because a pair of alleles has about 100 different values from one test, not just 15, starting from the pairs of 15 alleles. And if they're independent alleles, you can multiply the number of possibilities 100 times 100 times 100 times 100, 15 or 20 times, and you're out at a trillion or a trillion, which is a lot more possible human barcodes, these genotypes, from doing 20 tests on a person in one tube, than there are people. So they're very good at identifying people. This, by the way, was shown during the admissibility hearing and to the jury during this Wake. Oh, I didn't tell you about the Wakefield case. I apologize. So what happened, there's a young man who is strangled by a guitar amplifier cord. And John Wakefield's accused of killing him. There's DNA that's found. The lab chops the guitar amp cord into 3-foot sections, swabs them. The signs of a struggle, so they also swab the shirt and the victim's arm, anything that was in the apartment, to see if it's connected to the defendant. So that's the evidence that we're looking at. A picture on the left is a DNA mixture. It's by 2 strands of DNA instead of just one, say 2 people. The lab does what it does, whether it's a cheek swab or a 16-person mixture. It makes no difference. They amplify and separate out the DNA, and the data that we see in the center is what happens after the detection of the amplified DNA product. And instead of having 2 peaks, that would correspond to a person like a 10 and a 12, we see 3 peaks, a 10, 11, and a 12. This indicates that there's more than one person there, most likely. And the question is, how do we separate out the people? Here what the computer would do, it would separate out up to probability the first person. Suppose we know the first person's a 10, 12, then like a victim. Then it's looking for the unknown component, the unknown genotype. And there's more than one pair of alleles, more than one genotype that could explain the data when combined with the 10, 12. Again, I'm not going to go through everything I would do with the jury. The real punchline, though, is on the top right. There were 100 possibilities when we started. And when we're finished, there's only 3. That reduction of choice is where information comes from, actually of any kind, if you want to understand the concept of information in any field. And so instead of having the whole population with a small probability for 100 possibilities, it's now condensed to 3. Science will put probabilities to those numbers, and now this is an informative genotype from one of the 2 contributors. That's all done objectively, comparisons made, and then a match statistic is calculated. This is what the data actually looks like at one of the 15 locations in this case. On the horizontal axis, we have the different sizes. Those are the different lengths of DNA, how many times the word root was repeated. Vertically, that's the amount of signal detected from each of the amplified alleles. So we see a tall 15-19 pair on the outside. That corresponds to the victim in this case. We also see 2 unidentified peaks in the center of much lower height at around 100 on that vertical scale. That's 100 relative fluorescent units from the detection machine, RFU they're called. And the question is, with what reliability can we actually make some inference from this data about what are the genetic types of the people who left their DNA? So this is the problem that the crime labs ran into, and why the New York State Police couldn't analyze this, or most mixture cases, is they have thresholds and they throw out most of the data. In this case, they're keeping the victim's data taken from the victim. Of the 15 and the 19, it's not very probative. It's not telling us very much. And the peaks that don't correspond to the victim, that might tell us about who actually may have committed a crime, belong to a minor contributor, meaning he left a lot less DNA than the major contributor, the victim. And that's invisible to their analysis. It's just not there anymore. So there's no conclusion that they can make. The thresholds say there is a 15 and a 19 to their analysis, and then there's nothing more they can really say. They can identify the victim was part of himself. What the computer does is it starts with that data, and unlike human review, it doesn't touch the data. This is normal science. You don't touch the data. Evidence is evidence, right? You don't fabricate it, you don't discard it, you don't play with it, you don't Photoshop it. It's the data. And what the computer does is it explains the data, in this case, by making combinations of genotypes from 2 people in 100,000 different ways, of different amounts, different heights, different artifacts that aren't shown here, ways of explaining the data. In this example, there's a 15 and a 19 with 2 blue rectangles. That's the computer saying there's a certain amount of a 15 and a 19 going up to that height of 250. And now it's also proposing there's a second person shown in orange, a 17 and an 18. And what you see is that that pattern of 2 tall blue bars and 2 short orange bars on those alleles explain the data well. Decades ago, that might have been a place to stop, but with modern statistical computing, what the computers do is they try out many, many possibilities. The goal is not to say what's the best answer, it's to work out the probability of every possible answer, and do that objectively without knowing what the answer is supposed to be. And computers can do that. And so when it's done, of those 100 possible allele pairs at this one locus, most of the probability is concentrated on 3 different allele pairs, a 17-17, a 17-18, and a 1719. It turns out that 96% of the probability is concentrated just on that pair of minor peaks at the 1718 that you saw visually look like the most likely answer. The computer's not guessing the most likely answer. It's tried out 100,000 possibilities. And it keeps coming back to this one. How often? 96% of the time. That's how it gets the probability. So that's what an evidence genotype looks like from one locus from this evidence item, separated out from one of the two contributors. It's done objectively. It's not peeking at the defendant. We're not changing the data. Nothing like that's happening. The reason I'm showing you this picture is if we're going to explain a match statistic in court, I would like the trier of fact to visually see and understand to some extent what we're even talking about. It's not language. It's not a number. They're not going to understand that. But if they see a picture that reflects the math accurately, they can see the picture and say, I got it. So here's the picture. What a match statistic really is, it's a ratio of probabilities. It's the ratio of probabilities after seeing the data, shown in blue, divided by a random person before you saw the data, shown in brown. And what you see is that all this exists before you make a comparison. If you look on the bottom left, over there at a 1717, you see that coincidence, the brown bar is twice as high as the blue bar, which is the evidence. That would point away from somebody with a 1717 genotype. Same thing on the right with the 1719 and at the other 100 or so. But to get a match statistic, we focus in once we make the comparison, not before, but once we have the two probability distributions, after seeing evidence in blue and before in brown, and we say, what's the genotype of the person we're comparing with? In this case, it's a 1718. So we circled in red, attention is focused there. What's the ratio? Well, the ratio of the 96% in blue after seeing evidence divided by brown before seeing evidence gives us a ratio. The probability of an evidence match divided by the probability of coincidence. That's a match statistic. It's one of the dozen ways you can write it down. Probability after seeing evidence divided by probability before. And that gives us an 8. And doing that at all 15 locations, which are listed here on the left, the one we just looked at is at the bottom. The length of the bar is how big that number is. So some of them are out over 200, others are under 10. Since they're independent locations, they keep those lengths, those match statistics at each locus can be multiplied together to produce a result to answer the question, is the suspect in the evidence? And then, again, using the smallest number here, we can say that a match between one of the guitar amp chord segments and defendant John Wakefield is 300 times more probable than coincidence. What do we mean? We mean that picture, the match to the evidence in blue divided by coincidence in brown. And those are the words that reflect that picture. There's no gobbledygook here. It's just, that's what it is. It's a ratio of evidence match divided by random match. And then the victim, Brett Wentworth, is also present. This item collected from him, and that number is in the quadrillions. A quadrillion has 15 zeros in it. So hundreds of quadrillions has 17 zeros in it. And then using the number of zeros in the match statistic, this is what's in the report, and this is what's shown in court on pretty much the last slide. Here's a listing of the different evidence items, the swabs from the guitar chord, listed on the left, along with the shirt collar and where people think the fight might have happened with the two men struggling and John grabbing Brett by the shirt, as well as grabbing the victim's forearm. You can see on the left column 18 zeros. It's a billion. He's all over his own guitar chord, his own arm, his clothing. That's expected. But the second column, all the way on the right, these are the probative questions. When we compare the inferred unknown genotypes that were separated out from the evidence to defendant John Wakefield, what are the numbers we're getting? The number that we saw as 5 down, 8.48, is 8 zeros, so that number is in the hundreds of millions. And then we also see that his DNA is also on the shirt collar, and that's down on the bottom right, 18 zeros. That's a number of a billion. He's also not on other items. You see 4 down on the right, a negative 16. So that's 1 over 10 quadrillion. So he didn't leave his DNA in a detectable way on that part of the guitar chord. That's the statistics trying to explain DNA and the process in pictures and words, and not just coming in a white coat and pronouncing a number and leaving, which might not be helpful to a trier of fact. In this case, there was a Frye hearing, and one of the folders on the DVD has 12 subfolders of different categories. 20 admissibility rulings, background reading, applications in forensics. These folders can have anywhere from 3 to 30 documents in them, validation studies in different categories. These are the binders that are introduced at a hearing and introduced as exhibits, and depending on the interest of the court, either discussed briefly or discussed at more length. Probably the most important components are the validation studies. And there are 7 published validation studies in peer-reviewed journals starting in 2009. I'll show you pictures of a few of these from New York State later on, which makes them more interesting. The top 4 in the lighter shade of blue correspond to studies done on lab-synthesized data. These are good because you know what the answer is supposed to be, unless there was a lab error. They're made in the laboratory. You're making comparisons. The scientist, the expert, the manufacturer, the crime lab, whoever is bringing in evidence like this, they have done studies where they've synthesized mixtures in the laboratory of known composition, and then they do their experiments showing how well the statistical methods work. The bottom 3 are casework, and we did 2 with New York and 1 with Virginia like that. And the reason casework is important is because casework data often does not resemble the pristine data that you see from the laboratory. To use the vernacular, it looks like schmutz. It's just, you've got all this garbage all over the place, and simple programs, some of the European freeware programs, don't even work on real complex evidence, though they work very well on lab data. So we want to know both, and over the years we've devised methods that we and others use for being able to do reliable validation studies on actual casework, data from the field. I dropped all of the interesting pictures of scientific data, because after lunch it would have been a disaster, you all would have fallen asleep. But let me at least tell you what the main concepts are. You have these studies, you can look at them. The 3 main axes are, using the FBI's language from 20 years ago, sensitivity, specificity, and reproducibility. Sensitivity is asking, if somebody left their DNA, are you finding them? And then statistically, we can measure to what extent do we find someone who's actually there. Specificity asks, if somebody's not there, are we saying they're not there? And to what extent, statistically, are we excluding them? An exclusion isn't a verbal statement. Scientifically, it's a number. Are we strongly excluding people who aren't there? Numbers like 1 in a billion, or only weakly. Maybe some methods can't even exclude people, like the combined probability of inclusion that was most of the mixtures in the country for 20 years. So these experiments are done by comparing DNA or genotypes that are not in the samples against the evidence samples to determine what is the distribution, to what extent is somebody not, if they're not there, do we know they're not there? And it's done numerically. Reproducibility comes up because to solve a 100-dimensional probabilistic system of equations, you have to use random sampling methods. For judges who are elected, random sampling methods are really clear. It's like polling. If you poll your district and you poll it week after week, I don't know how frequent, I've never run for judge, I don't know how often you poll, but you expect to get some possible error from the sample, and every time you sample, the number will be a little bit different. If it's always the same, there's something wrong, right? Somebody's not really sampling. So these methods are like random sampling, but instead of a population of voters, it's of maybe 100 variables. If the methods are reliable, the polling numbers will be similar every time, but not identical. The same thing's true with reproducibility studies, with Markov chain Monte Carlo, which is a way of solving the equations. There's two different ideas. One is what are the equations that are in the software? That's the main event. And then secondly, those equations together with the data need to be solved. That's called MCMC, or Markov chain Monte Carlo, and it's the standard way in science of solving high-dimensional interlocking equations. And so we measure that. If we got 10 zeros after the 1, 10 billion the first time, do we get 1 billion or 100 billion the next time? Or do we get 2? And if the system is reproducible, the numbers are usually within a factor of 10. So that's measured, graphed, plotted, tabulated, analyzed statistically, and so on. So again, the three ideas: sensitivity, we're finding people who are there; specificity, we're showing people aren't there who aren't there; and reproducibility, when you run the program several times, are the numbers close? There are a lot of indicia of general acceptance. We have a whole binder on just who's using it, what they're using it for, different projects, how it's used for applications that have nothing to do with criminal justice, like mass disasters or a scientific tool in the laboratory. And this just goes through a 20-year history of where we are with TrueAllele, which is, we're not doing a hearing on TrueAllele today, so don't worry about that. This slide was actually prepared by New York State Police many years ago, which is why it's so animated. I don't prepare animated slides. And I think it was prepared by Dr. Barry Duesman, and it shows an event 12 years ago when the New York State Police were using our reference sample system, the one that eliminated the backlog of buccal swabs in England and did 350,000 samples. A whole other story. Basically, get rid of a building full of 100 people, replace it with computers. England was happy, but not the 100 people who used to be in the building. And so at the peak of the backlog of the DNA database in Albany, they were up at over 55,000 backlog samples. And within, a year of having brought TrueAllele on board, not the casework system that does all the statistical stuff, but a predecessor that was more for reference samples, it had dropped to under 5,000. And they wrote papers about this. And it was the first large-scale use of computing that could reallocate resources in a crime lab in the US this way. As I mentioned, we reanalyzed the World Trade Center DNA data. If I were doing a Frye hearing in New York State, I'd certainly mention the World Trade Center. We were writing papers with them that described results. You have both of these papers. I'm going to just tell you about one of the figures there, because these two papers have results that are complementary. What you're seeing on the x-axis horizontally are 8 different reported cases from the New York State Police Albany Forensic Identification Center. The orange bar is showing the match statistic that they reported using their inclusion method, CPI. The blue bar is showing what TrueAllele found on the identical data in an automated way without people. And the heights of the bars, the average of the orange bars is 7 zeros is around 10 million. And the height of the blue bars is more match information, which was 13 zeros, or 10 trillion. The difference is 6 zeros. So what this was showing, as a very early validation study for these systems of probabilistic genotyping, was that on all these cases where the New York State Police reported a match statistic, the computer was getting, on average, 1 million times more information. It's actually later on we realized that the computer was showing information, and if you look at the brown bars, it's just, they're always reporting around 7 zeros. It's always 1 million or 10 million. But we didn't realize that till later on. Okay, so it preserves more evidence. This is a second paper, much more extensive. We looked at over 85 genotypes across many cases, 2- and 3-person mixtures, a very long validation paper. And this was an amazing slide that we showed at the DNA subcommittee of the Forensic Science Commission in New York. What you're seeing on the x-axis is just 85 cases, and that blue cloud is showing in decreasing order the match statistics of what the computer found. So it starts up at around 20 zeros and then moves over, falls down. The average is around 15 zeros or a quadrillion. The colored bars are what human review reported in these cases, actual issued case reports that you would have seen in court. And you can notice that with these different methods they used, I won't go into the technical details of the gray, the blue, and the orange, that generally the numbers are smaller, but that wasn't what was profound. What was interesting is that when the computer found an answer on these cases of what now we consider to be very easy mixtures, they're just 2- or 3-person mixtures, 70% of the time the lab failed to give any answer at all. And that was normal across the studies we did across the country. That inconclusive was the main result we got from informative data that computers could reliably analyze. In fact, we were doing estimates at the time. I think at this point, my estimate, based on the numbers from the New York State lab after 10 years, is probably about 10,000 cases where they reported inconclusive instead of an actual result. And that's just from one lab in New York. This was something that Suffolk County showed on their own validation study at the DNA subcommittee. Here, only what you're seeing on the right half did the lab have a result and TrueAllele. On the left, there's no lab result, it's just TrueAllele. So in the Suffolk lab, more than half of the informative mixtures were not being reported 10 years ago. And based on many, many validation studies and whatever was asked for in presentations, in 2011, the DNA subcommittee and then the full Forensic Science Commission approved TrueAllele for casework in New York State based on validated science. Probably worth mentioning that there has never been a validation study of the method that's regularly used called combined probability of inclusion, so no one's even bothered to have a Frye hearing on it. Even though it's not believed to be reliable, it's what you see and people just accept it. This was the ruling from Judge Kokoma. In the Wakefield case, there was a lot of testimony from people in the laboratory. At New York State Police had done their own independent studies out to 4-person mixtures. Just a lot of science presented in this Schenectady hearing. I think I provided you with this ruling as well. This was a recent ruling that was interesting. The first case I ever testified in New York State was in Chemung County in Elmira, and it involved a serial rapist who would sort of chat up his girlfriend's friends and then come back and rape them. And he was hard to catch, and at some point he left a pair of purple gloves as he was in a grainy way being caught on a bicycle leaving an apartment, and the gloves were found. He had a very strange modus operandi. It's before lunch. I won't go into it. Kind of gross. Anyway, there was a lot of DNA on the gloves from the victim. He admitted they were his gloves. The victim's DNA is on them. His DNA is on them. And he was convicted. And then 4 or 5 years later, his attorney on appeal successfully got a Frye hearing, a pre-trial hearing 5 years after the fact. I've never heard of it. Maybe you do it routinely, but I just never heard of it. My understanding of the word pre is that it means it's before the trial. So we did a post-trial Frye hearing. It was earlier this year, and we had to roll the clock back, right? Kind of like Back to the Future. We had to do everything as it was at the time. And so that was an interesting hearing. And so we'd introduce evidence. We couldn't talk about much of it. So this is another ruling from Chemung County this year where TrueAllele was accepted as reliable. Cybergenetics has testified in 14 New York TrueAllele trials where TrueAllele evidence was introduced, maybe about a half dozen different counties, all sorts of offenses. I testified in the first 7, and since then, analysts from cybergenetics have been coming out to Suffolk County, New York County, wherever, and giving testimony. There's some interesting cases here. I mentioned Casey Wilson with the gloves and John Wakefield. The case with Adam Mogan was interesting. We had several cases like this. The one in Westchester was similar but most interesting. There was a man who regularly had sexual intercourse with his daughter every week from age 7 to age 14. And there was all this evidence of DNA on the blankets, mixtures all over the place of his DNA and her DNA. And of course, it was inconclusive. That's why I say inconclusive harms criminal justice. And using TrueAllele, we were able to separate out the mixtures. I think that led to a plea agreement in the end, and the girls moved on. So incest rate can be a problem, and DNA can help resolve that. There are other cases. One of them is for the defense, and I wanted to tell you about that one because this is something that comes up a lot, and we see it. If you work with a lot of prosecutors, the way I do, because the way our process works is that we do a free screening so that we're not binding anybody to anything. They send us the data from the lab, whether they're prosecution, police, defender, innocent, civil, whoever they are. And then we take a quick look in TrueAllele. We send back an initial summary of what we found. For example, if we find the defendant is all over the DNA, they're not going to order a report. If it's a prosecutor or police and their guy isn't there, they're not going to order a report. So that's how we start. We started off working mainly with prosecutors. We always had the sense, there's the demonization of the opponent. But what we found over time is that lawyers like to win. Maybe you've seen that, I don't know. And so there's no difference, really. I mean, theoretically, I've worked with some amazingly ethical prosecutors, amazingly ethical defenders, and it always comes as a surprise to us when somebody who's a prosecutor who uses DNA regularly is banging on us and attacking DNA. It's like, why are you doing this? This is a tool you use. We don't really get it, but we deal with it. So this was a case that came to us from the Legal Aid Society, and it was a case, Stephenson, of a man accused of pushing a woman onto the subway tracks. She was injured but rescued before any train arrived. And there were DNA stains on her dress, and the prosecution wanted to use the DNA to show that he was included and he was the guy who did it. So what OCME came back with were 3 exclusionary items and 3 inconclusives. Legal Aid sent us the inconclusives. We looked at that, and it turns out they were exclusionary too. If you used the right software, he wasn't there. So I think these are the results, right? If you look on the right side, they're all in red, so that's exclusionary. The numbers are octillion, septillion, and hundreds of thousands. So statistically, he's just not there. And so we thought, I just go to court, and okay, he's not there. But that isn't what happened. I looked over the transcript, and it was really aggressive. And we asked people afterwards, why would somebody care so much that somebody's actually excluded? And statistically, we showed it with science. And this is what we heard, and this is what we discussed with people, and we've seen. There's a chain of logic that can happen in a courtroom. And I've seen it in other cases, particularly if I've testified for the defense, which I've done a few times. It goes something like this. The results are inconclusive. Therefore, the defendant cannot be excluded. Logically, the argument will continue, maybe during closing. The defendant is included, because if they're not excluded, they must be included. And if they're included, that means their DNA is there and they're guilty. And I've discussed this with a lot of people around the country, and almost all people who testify have seen this in courtrooms and defenders. I personally, as a scientist, I find it horrific. How can you take nothing, zero, like you never collected the DNA in the first place? It's inconclusive. And you're turning it into guilt. As a scientist, I'm just staggered by it. But that's what someone was trying to do in this case. But in fact, Stephenson wasn't on any of the items, regardless of the legal arguments. The Johnny Lee Gates case is nationally known as a death penalty case in Georgia. There's a mentally handicapped African-American man who's accused of raping and killing a white woman who is the wife of a serviceman down in Georgia. It's a death penalty case, and over time it became clear that there really wasn't any evidence that wasn't planted. It became clear that his knowledge of the crime scene was because they had walked him through before, but didn't tell the prosecution, but hadn't told anybody. That his fingerprints found on the radiator was when he came back to be interviewed. So there really wasn't actual evidence. It also turned out, and this is why the case was, if you look up the name online, was notorious, is that this was at a time of racial bias in jury selection. And as was later shown by the Innocence Project, over a 5-year period, there was tremendous bias with racial letters indicated next to jurors' names and largely all-white juries convicting these people and sentencing them to death. The Innocence Project came across evidence that nobody thought existed anymore, and TrueAllele was able to look at this evidence, and this was the evidence that was actually the ligatures used to bind the victim and strangle her. And they were 4-person mixtures, and as you see in the red on the right, TrueAllele excluded Johnny Lee Gates from all of these items. Here again, the prosecution was not that happy letting this man go free after 40 years. And he kept his case alive. I mean, he may have been mentally handicapped, but he knew how to keep writing to the judge and not making legal arguments, but at least protesting his innocence. And during my cross-exam last year, the prosecution argued DNA transfer, something I lecture on, and made an argument I don't think anyone's ever heard of, which is the reason Johnny Lee Gates' DNA was not on these 4-person mixtures where we found information that human review could not find, anything, someone there or not there, but we could show exclusion, was because it fell off. I'll tell you what I told the judge, because you'd be interested. You want to hear? So the Innocence Group, Claire Gilbert, actually the director, said, can I come up with some analogy that the judge would understand? The judge was over 70, there's a lot of science going around, all these arguments. What does it mean DNA doesn't fall off? So I had thought about it the night before and tried it out on her. And she asked me, could you explain to the judge about your thoughts on DNA falling off? And it was in Georgia, so I explained, Your Honor, if you have, because when you're doing a hearing, you're looking at the judge the whole time. It hurts your neck, but you're looking at the judge. You know, we've got this, so imagine you have a cup of coffee and you add some sugar. It's the South, you add some more sugar, right? It's sweet, everything's sweet down there. And you stir in the sugar. And now you add some cream, right? You add more stuff to it and you stir it in. It's still sweet. The sugar didn't fall off. And actually, people said that was when he got it, that this was just silly. Whatever the argument was, that DNA fell off. But it's an amazing order. It's worth reading. He goes through, it's posted everywhere online. If you look for Johnny Lee Gates and the order, I think they're having more hearings about it. The judge ordered a new trial. And he talks about the jury bias, the planting of evidence, and the only thing that he could bring in under the law was newly discovered DNA that was exclusionary. Because on everything else, the time limits were statute of limitations had run out. So this is another article that I wrote. I didn't give it to you, you can find it online. And this is something that I observed in court where experts, and it happened again in a trial in Florida when I was out of town. I think it happened because I was out of the country, and so they could bring in an opposition expert who just snuck this in. What happens when you have really good match statistics, but you have a lawyer or an opposing expert who distorts the match statistics to persuade a jury that something that makes no mathematical sense casts doubt? And so this paper gave 3 examples of how that would work based on a talk I gave at the American Academy of Forensic Science meeting the year before. And it shows how it's done with testimony. Here's one example. This is the first of the 3 ploys. We have probabilities of different genotypes. Now, the probability is the probability, and then the argument gets made, but look, the defendant, shown in red, is 24%, but there's this other genotype on the right. It's got more probability. It's 28%. So how could it be him if something else has more probability? It's kind of like asking if the weatherman predicted sunshine and it rained on your picnic. How is that even possible? Because you know it's 28% chance of sunshine. But the fallacy here, and here's actual test. I'll read the bottom 2 lines. The defender says at D13 locus, we're going to go through these loci, and the defense expert says it was the second highest. This one listed in the table is the second most probable. He's not lying. It's true. It's just misleading for several reasons. The first is a match statistic is a ratio, as you heard half an hour ago, between evidence probability and coincidence. It's not looking at each one separately. And in this case, the ratio of what was relevant of 24% divided by 3% was a match statistic of 8. What's going on elsewhere doesn't make any difference. It's just not relevant to the likelihood ratio. So it's a true statement that there could be higher probabilities, but it's the ratio that matters for the defendant. And the way the argument went is the prosecutor objected based on relevance, and the court sustained that. It's not just trial attorneys who might try to distort evidence. There's a lot of stuff from the federal government that doesn't seem like science to a lot of practitioners. Suppose your match statistic is only 103. Well, 103 doesn't sound that persuasive. And in fact, Professor Jay Kohler at Northwestern has done studies that with match statistics under 1 million, juries may not be persuaded. So why give a number less than 1 million when you can substitute words and hide the number from the jury? That's new federal policy. These are called verbal equivalents. Why tell the jury 103 when you can look it up on your table and you can say, there's moderate support for a match? And when asked about it on cross, well, that's my protocol. I can't tell you the number. I can just tell you there's moderate support. But giving a number of 103 when there's 200 people in the courthouse isn't that probative. So I think people are playing games with this. And you don't need to do it either. There are ways of reporting error. Like what's the chance that somebody who didn't leave their DNA would be as strong against an innocent person? And computers can calculate that. I wanted to show you the slide from Allegheny County because we've done 75 cases there. They have 15 trials, 8 acquittals or exonerations. The column called outcome, second from the right. This is important for judges. The main outcome when computers bring reliable DNA evidence back into a case after the lab has thrown it out is a guilty plea, because now the DNA is back. If there was no DNA, then there may have not been strong evidence. But if there is DNA, what we've seen is about half the time we don't go to trial. Sometimes we find out about it as we're on the way to the airport. Sometimes we find out about it waiting outside the courtroom, but usually in a different state. When you bring evidence back into criminal justice, you can get better negotiated results than if the evidence isn't there at all. And I'll just leave you with a thought. This is something that I'd like to see happen. I don't know. I know other people would like to see it happen. If we know that hundreds of thousands, maybe millions of mixture items have been misinterpreted, called inconclusive, wrong match statistics given when they're actually even given, 10 or 20,000 just in New York State, then if you have an automated machine that can just take the data in and spit out numbers, why not go back and find out what actually happened and who may not deserve to be in jail or who may actually deserve to be in jail. So with that, thank you very much.